And so today we're going to do the catalase experiment and nitrate reduction experiments. Here we're going to talk about them. So we have already talked about catalase in lecture. So you're familiar with that particular enzyme. What does catalase break down? What does catalase break down? Have we forgotten already? So what does catalase do? Yeah, I didn't get that. It breaks down. I gotta talk to you. Yeah, it breaks down hydrogen peroxide. Remember, hydrogen peroxide is very toxic, causes free radicals to form, it can lead to a lot of damage. But it is produced when superoxides are broken down, right, by the enzyme superoxide dismutase. And so we actually produce a lot of hydrogen peroxide. So a catalase is an enzyme that can break it down. When it breaks down catalase, it produces water and oxygen gas. So the way we can see this in lab is by bubbles, like bubbles of oxygen gas. So what kinds of organisms do you think would produce catalase based on oxygen requirement? What kinds of organisms?
Would it be anaerobes?
What kind of anaerobes? Right, because obligate anaerobes would be killed by oxygen. But other anaerobes would be okay. Like what?
Obligate anaerobes?
No, those are going to be killed.
Oh, facultative?
Right, facultative anaerobes, aerotolerant anaerobes, anything that lives in oxygen, whether or not it's using it, right? Need to have these enzymes to deal with it. And of course, obligate aerobes, right, because they need oxygen, so they need to have catalase as well. So we can use catalase to help differentiate different types of gram-positive cocci. So Staphylococcus are all catalase positive. Streptococcus and Enterococcus are catalase negative. When it comes to Clostridium, that's typically catalase negative because we know it doesn't like oxygen. And then Bacillus, they are catalase positive. But when it comes to using Bacillus, remember that Bacillus makes spores, right? So in a slant culture, in order to make sure you get the best results, you want to take your sample from the bottom of the agar slant, where the bacteria are young and healthy. If you take your sample from the top, it might all be spores, which don't have catalase. So, when using Bacillus, again, think about what you remember about Bacillus on an agar slant and where would be the best place to get your sample. Right, because Bacillus is catalase positive, but if you have a lot of spores, you're not going to see that. It can be used on Mycobacterium, but we don't actually test for that in lab. So let's watch a video.
Writing's not that easy, but Grammarly can.
video, we're going to discuss the theory behind the catalase test. All right, catalase is an enzyme that detoxifies hydrogen peroxide into water and molecular oxygen, as you can see in this reaction right here. Hydrogen peroxide is actually a dangerous molecule in any organism because it's prone to forming radicals, which can actually damage DNA and proteins and lipids. And the catalase test is one of the most important tests in differentiating between the staphylococci, which are catalase producers, and streptococci, which do not produce catalase. Okay, so that's a key here. We can use this to distinguish between staphylococcus and streptococcus. Okay, so when catalase disproportionates, which is just a type of reaction, when it disproportionates hydrogen peroxide into water and molecular oxygen, it turns out the oxygen is actually a gaseous molecule, and it will actually bubble off. In fact, humans make catalase. We actually express this enzyme. You should do an experiment next time you skin your knee on the ground. What do most people do when you skin your knee on the ground? You take some hydrogen peroxide, it's usually in those brown bottles, right? You pour some, usually either pour it or put it on a cotton ball, put it on the wound. And I guarantee you what you'll see next time you do that is you'll see bubbles produced. Why? Because your body produces catalase to protect from hydrogen peroxide that's naturally produced in your body. But if you take some from the outside environment, like from the bottle, it'll still produce those oxygen bubbles and you'll be able to see those. So therefore, the key with the catalase test is the catalase test is positive if bubbles are observed after adding the hydrogen peroxide. Okay, so a fairly simple result to observe. This is kind of what they should look like. If bacteria express catalase, and an example of that would be Staphylococcus, then oxygen bubbles are going to be evolved. As you can see here on the top right here, you can clearly see some bubbles. You can see them down here also for that catalase positive result. Okay? If it's catalase negative, such as right here or on the bottom of the top picture, such as Streptococcus, then no catalase is expressed or made by the bacteria, and those results are going to be catalase negative. Okay, so make sure to watch the demonstration on the catalase test.
Now that we've helped you optimize and save with Liberty Mutual, we can finally do some beach reading.
So we're looking for bubbles. Now, as he said, if you have a cut or a scrape and you put peroxide on it, it will bubble like crazy because we're very aerobic organisms. Our blood and tissues have a lot of catalase. And a catalase is actually a very tough enzyme because I once had, some cloth that had some dried blood on it, and I put some peroxide on it, and it still bubbled like crazy. So, catalase is a tough enzyme. And in the slide test procedure that we're going to look at, you can either put a drop of catalase on the slide first and then put your loop full of bacteria into it, or you can smear your bacteria onto the dry slide, even let the bacteria dry out, and then put a drop of peroxide on top, and it still bubbles because catalase is a very tough enzyme. And so in lab, what we would do is each group would get 2 glass slides. You would put 2 separate drops of hydrogen peroxide on each slide because we have 4 organisms we're testing. And then get a loop full of those organisms, put them in the drop, and see if it bubbles. Record your result, and then you would discard your slides into the dish of disinfectant, right? Because we don't put glass slides in plastic bags, so not the biohazard bags, but we would have a dish of disinfectant where you could put those slides. Since they were wet slides, it's, following standard procedure. And then, as I mentioned for bacillus, it's important to get your sample from the bottom of the slant where the cells are young and healthy. So we'll watch another video, which again talks about the procedure. It shows the slide test.
I still don't understand why she want to go out with me.
I don't either.
And what it is if an organism is catalase positive, we're going to be doing the catalase test. And what it is if an organism is catalase positive, it can break down hydrogen peroxide into water and oxygen. So a catalase positive reaction is going to cause bubbles, whereas a catalase negative reaction is not going to have any bubbles at all. So what I did was I labeled the organisms out of the first grouping of organisms we use in class, which are positive, and those that are negative in regard to gram-positive cocci. So we have the Staphylococcus and the Micrococcus that should be positive, and the Enterococcus and the Streptococcus, which should be catalase negative. Now, if you look at the grand scheme of things, most everything is catalase positive except for Strep and Enterococcus. But in the first grouping, we have the gram-positive cocci, gram-negative cocci, and gram-negative cocobacilli. So that would be Staphylococcus, Micrococcus, Enterococcus, and Streptococcus are all gram-positive cocci. Then we have Neisseria, which is gram-negative cocci, and Moraxella clateralis, which is the only gram-negative cocci in Moraxella, and Haemophilus, which is gram-negative cocobacilli. So I put an organism on each that should be one that should be positive on here. So I put Staphylococcus aureus on here, and I put Streptococcus agalactiae on this one. So I'm going to add my hydrogen peroxide and look for bubbles. Okay, so as expected, the Streptococcus does not show any bubbles. On the video, it looks really white, but there aren't any bubbles. Whereas on the Staphylococcus aureus, look at all those bubbles. Okay, so this is a positive test here for Staph, and this is a negative test here for Strep.
Muscle milk helps build muscles. With 40 grams of protein.
So we would test Staph aureus, Micrococcus luteus, some species of Bacillus, either Cereus or Subtilis, and Enterococcus faecalis. So I want to relate the catalase result with the oxygen requirements. So you should be familiar with some of the oxygen requirements of some of these organisms. But Staphylococcus aureus, as you saw in the video, as we explained, has what catalase result?
Positive result.
It's positive, right?
It's positive. Yeah.
And so what you don't know that Staph aureus is a facultative anaerobe. So that's its oxygen requirement. It's a facultative anaerobe. So remember, facultative anaerobes love oxygen, but they do not need it. So no surprises with Staph aureus. It's catalase positive. Micrococcus luteus, do you remember the oxygen requirement for Micrococcus luteus? What's the oxygen requirement for Micrococcus luteus? We did that lab a couple of weeks ago, right?
Obligate error.
Right, it's an obligate aerobe. So what would you predict the catalase result to be?
Negative.
It's an obligate aerobe?
They still requires energy. Okay, positive.
All right, positive. So Micrococcus luteus is positive for catalase. Bacillus cereus. Christiana, can you turn your microphone off? So a Bacillus cereus or subtilis, either one. We did use Bacillus in the oxygen lab, and what was its oxygen requirement?
Was it a facultative anaerobe?
Yes, it was. So what would you predict its catalase reaction to be?
Positive?
Positive, right? It is. And then there's Enterococcus faecalis, which I'll tell you is another facultative anaerobe. So what would you predict its catalase result to be?
Positive?
Right, positive. But in fact, it's negative. So we've just seen that Staph aureus and Bacillus are facultative anaerobes, and they're catalase positive. But Enterococcus is also a facultative anaerobe, but it's catalase negative. How can that be? It loves oxygen. Huh?
Is it because it's a gram-negative bacteria?
Oh no, it's gram-positive. It's a gram-positive coccus. And it loves oxygen, but it's catalase negative. So how can that be? So Enterococcus faecalis is a facultative anaerobe that is catalase negative. So how can that be? Because it loves oxygen. It makes lots of toxic hydrogen peroxide, so how can it survive? Nobody has any ideas?
No, I can't think of anything.
Gee, how quickly you guys forget. Remember, we talked about oxygen requirements, and we talked about enzymes like superoxide dismutase, catalase, and what's the third one? Nobody remembers the third one? An enzyme that breaks down hydrogen peroxide.
Oh, is it peroxidase?
Yep, peroxidase. So remember, there's two enzymes which can break down hydrogen peroxide, but peroxidase just produces water. So there's no way to test for that. But since I'm telling you that Enterococcus is a facultative anaerobe that loves oxygen and is catalase negative, you must assume that it has peroxidase. So does that make sense?
Yeah, it does.
So any questions on catalase before we move on?
So, Professor, is it because it contains peroxidase? That's why it's
Yep.
negative? Okay.
Yep.
Thank you.
So for nitrate reduction, So some bacteria have the ability to use inorganic compounds as final electron acceptors in anaerobic respiration. So remember, we talked a lot about aerobic respiration. It's aerobic because oxygen is the final electron acceptor. But we also mentioned there are anaerobic pathways that can use inorganic compounds like nitrates as a final electron acceptor. So we're going to be testing to see if bacteria have the ability to reduce nitrate into nitrite. So from 8 to 8, right? NO3 to NO2. And if bacteria can reduce nitrate into nitrite, that means they have the enzyme nitrate reductase. Now, in general, for biochemical tests, we're trying to determine if an enzyme is present. And we know the enzyme is present if we find the end product of that enzymatic reaction, right? So for catalase, the end product was oxygen gas, right? We saw bubbles. That's how we knew catalase was present. So for nitrate reductase, we're going to determine if nitrites are present. So remember, the bacteria use the enzyme nitrate reductase to reduce nitrate into nitrite. So we're going to test for nitrites and see if they are there. Now, the way that we do that is by using two chemicals called sulfonylic acid and alpha-naphthylamine, which we call nitrate reagent A and nitrate reagent B. And reagent is just a fancy name for chemical. So we have two chemicals, A and B, sulfonylic acid and alpha-naphthylamine. They react with nitrite to form a red color. So if you inoculate your bacteria, you let them grow, and then in the next class you add the two chemicals, nitrate A and B. If it turns red, that's a positive result. Now, in general for biochemical tests, what I just said, you have to inoculate, incubate, then you perform the test. And that's how that works for almost all the biochemical tests. And I just bring this up, even though we're not going to be doing lab face-to-face. When you're doing your unknowns, which are coming up the week after next, in lab, students would inoculate all the different biochemical tests. And a common problem is that students will inoculate, the test medium with bacteria and then immediately add the chemicals, to look for a reaction. Well, if you've just inoculated, the bacteria haven't had time to grow and do anything, right? And so sometimes students forget that after you inoculate, you have to incubate, let the bacteria grow, and if they're going to make a product, you have to give them time to make that, and then you test in the next lab period. So it's something that I would have explained to students, face-to-face, but I just want to give you that same experience that when doing biochemical tests for almost everything, you have to let the bacteria grow first, so they can do something, and then you test to see if they did something. So we're using nitrate broth. So it's a nutrient medium that contains nitrates. So if the bacteria can reduce nitrates into nitrite, we'll be able to see that when we add our two chemicals, nitrate A and nitrate B. Now, if we add nitrate A and B and it turns red, that's a positive test result, and you're done with that tube, because it turned red. But maybe you add the nitrate A and B and there's no red color. So then you will add zinc. So zinc is a metal, and you're just going to add a little tiny pinch of zinc, rotate the tube between your palms for a couple minutes, and see if there's any color change. And if a red color develops after zinc, that's a negative test result. And the reason why that's a negative is because zinc reduced nitrates that were still there into nitrites. So now it turns red, because we already have the nitrate A and B chemicals, they're there. But initially there was no color. You add zinc, now it turns red. So what that means is is that the nitrates were still there. The bacteria had not touched them, right? Because the bacteria do not have nitrate reductase. But zinc is a reducing agent. Zinc can reduce nitrate into nitrite. So now the tube turns red because nitrites were produced by the zinc. But since zinc is doing it, it's a negative test result. And then sometimes you add the zinc and there's still no red color. So you added nitrate A and B, no red. You add the zinc, no red. It's a positive test result. And the reason why is because you added the two chemicals, nitrate A and B, which tests for the presence of nitrite, and there were none. Then you add zinc, which can reduce nitrate into nitrite. So zinc is basically testing for the presence of nitrates. But you add the zinc, still no red color, so there were no nitrates either. So all the nitrates are gone, all the nitrites are gone, but there's nothing left to turn red. So where did everything go? It turned into gas, or something like nitrogen gas, ammonia gas, nitrous oxide. So some type of gas gets produced, usually ammonia or nitrogen gas. And so if there's gas produced, that's still considered a positive result. And the reason why is because you cannot get to the end of the pathway unless you start at the beginning. And so cells have to start by reducing nitrate into nitrite. But then the nitrite is further reduced all the way down to some type of gas. So no color after zinc is a positive test result because all the nitrates have been reduced, all the nitrites have been reduced, everything's been reduced down to a gas. And we include a little fermentation tube inside of the nitrate broth tube, our little bubble catcher, and you would see a gas bubble inside that tube, which was also a clue that you had what's called a super reducer. It has nitrate reductase, but then it continues with other enzymes to reduce everything down to a gas like nitrogen gas. So for this test, the easy way to remember is if it turns red before zinc, it's positive. If it turns red after zinc, it's negative. And if there's no color after zinc, it's positive. So red before zinc, positive, red after zinc, negative. No color after zinc is positive. So does everybody follow that?
Yeah.
So we give the bacteria nitrates. We want to see if the bacteria have the enzyme nitrate reductase, which reduces nitrate into nitrite. Nitrite is the end product we test for. We test for nitrite by adding nitrate reagent A and nitrate reagent B. And if nitrites are present, a red color develops. So that's a positive test result. If no red color develops, we add zinc. If nitrates are still there, untouched by the bacteria, then the zinc will reduce the nitrate into nitrite. Now the tube turns red because the zinc made nitrites, not the bacteria, so it's a negative test result. And if you add zinc and there's still no red color, that means there's no nitrates, no nitrites, it's all been reduced, which is still a positive test result because you have to start with nitrate reductase to get to the very end. And since we're testing for nitrate reductase, no color after zinc is a positive test result. So questions about that? So we'll watch a little video actually showing some tubes and a guy adding some of the chemicals.
Hello folks, today we're going to talk about the nitrate reductase test. And so we're looking for the ability of a microorganism to break down nitrate to nitrite. And then even, you might be able to break it down even further to a gas, and the gas would be either nitrous oxide, nitrogen, or ammonia. And so here I have 3 different organisms, and I'm going to have to add 3 different reagents to the tubes. 2 if it's positive for the first step. So the first step is looking for nitrate reduced to nitrite. And so that's going to be something that E. coli does. And so this is E. coli. I'm going to go ahead and mix it up just a little bit. Then I'm going to add a few drops of sulfonylic acid. So sulfonylic acid, I'll just add about 5 drops or so. And then another reagent, which is alpha naphthalamine, and I will add a few drops of that, and there should be an immediate red color develop. So you can see that red color, and this is positive for the reduction of nitrate to nitrite. And this, if you do this, and this happens, and you have this result, nitrate to nitrite, after the addition of 2 different reagents, sulfonylic acid and naphthalamine, then you're done, and you don't have to do anything else. Right? You should take your results down pretty quickly, because this will oxidize again and turn amber again. Okay? I'm going to show you a negative test. And so with the negative test, we're going to use Enterococcus faecalis. So if I take Enterococcus faecalis, mix it up a little bit, make sure it's nicely mixed. Take that and add about 5 drops of sulfonylic acid, 5 drops of naphthalamine. Nothing will happen. So I'm going to add this in here, and you can see nothing happens. Right? So at this point, 1 or 2 things has happened, and we're going to have to figure out what we need to do to establish what happened. First thing that might happen is nitrate is still there in its pure form, and it didn't get reduced at all because the microorganism did not have the enzyme nitrate reductase to break down or to hydrolyze or to reduce nitrate to nitrite or to a gas. Right? So what we can do to do that is we can just simply add a little bit of zinc. And so I'm getting a toothpick here, and I'm going to take a little bit of zinc dust, and I'm going to add that little bit of zinc dust to the test, and I'm going to shake it up a little bit. And if nitrate is still there in its pure form, zinc will reduce it immediately, and because the other reagents are in there, you're going to get a red color develop. So after the addition of zinc, if you get a red color, and that means nitrate was there in its pure form and it didn't get reduced, we reduced it chemically, but that means the test is negative for the organism that you're testing. Okay? The last type of result we can have is we can have an organism that reduces nitrate to nitrite, but continues to reduce it to a gas, and those gases can be nitrous oxide, nitrogen, or ammonia. And we have no way to test for which one, but we can test to see whether or not nitrate is still present in the tube. And so it's real easy to do. Again, this is Pseudomonas aeruginosa, which is a big reducer of nitrate. It takes it to a gas. And so again, we'll add a few drops of sulfonylic acid, a few drops of naphthalamine, and nothing more happened, right? So nothing happened, right? So again, that's step 1. And with step 1, if nothing happens, then you have to go to step 2. And so in step 2, you're going to add, again, some zinc. So take some zinc. And mix it in. And if you recall, when we added zinc to the Enterococcus faecalis that was negative, after a few shakes it turned red. Here, no matter how long I vortex it, it's never going to turn red because nitrate has been reduced to nitrite and further reduced to a gas. We don't know which one, nitrous oxide, nitrogen, or ammonia. And we just don't have the ability to figure it out. But we can figure out that nitrate is not there in its pure form. Therefore, it's been reduced to a gas. So after the addition of zinc, if you get no red color, then that's positive for the reduction of nitrate to a gas. Okay?
It's kind of funny how he threw the whole toothpick in there. We don't do it like that. We just use a little spatula to add some zinc. And he was also using Pseudomonas aeruginosa, which is a pathogen, so we don't use that either. We use Pseudomonas fluorescens instead. So remember, the main idea, we're trying to determine if the bacteria have the enzyme nitrate reductase. We do that by looking for the end product, which is nitrite. We test for nitrite by adding two chemicals, sulfonilic acid, or nitrate A, and alpha-naphthylamine, which is nitrate B, at equal amounts to the tube. If a red color develops, that's a positive test result for nitrate reductase because nitrites are present. If no red color develops, we add zinc. Zinc is basically testing for the presence of nitrate, because if nitrates are still there, the zinc will reduce them into nitrite. Now the tube turns red, but the zinc did the reducing, so it's a negative test result. If we add zinc and there's still no red color, that means there's no nitrates, it's all been reduced to some type of gas, usually ammonia or nitrogen, but could be nitrous oxide or nitric oxide. And we include a little fermentation tube in our tube so you can see that gas bubble. And we call such an organism that reduces all the way to a gas a super reducer. So here are the organisms that we use, and I've indicated whether or not we've added those chemicals and what the result is. So for E. coli, we add nitrate A and B, and it turns red right away. What does that mean?
It's positive.
Right, it's positive for nitrate reductase because the red color indicates nitrites are present, right? So the bacteria reduced nitrate into nitrite using the enzyme nitrate reductase. Then Micrococcus luteus, we add nitrate A and B, no red color. So then we add zinc. Now it turns red. So what does that mean?
It's negative because the zinc made it turn red.
Right.
The bacteria.
Right. It's a negative result because the zinc did their reducing. So the nitrates were still there, not touched by the bacteria. They're still there, and zinc reduced them into nitrite. Now it turns red because the chemicals A and B were already added. And in Pseudomonas fluorescens, we add nitrate A and B, no red color. We add zinc, still no red color. So what does that mean?
Positive.
Right, it's positive. Again, because all the nitrates are gone, all the nitrites are gone, everything's been reduced to some form of gas. But you can't get to the gas unless you start at the beginning with nitrate reductase. So red before zinc, positive. Red after zinc, negative. No color after zinc, positive. So any questions on the nitrate test?
No, but I have a question about nitrates in like deli.
Uh-huh.
What is it? Is it this kind of nitrate? Is it this?
They're preservatives.
Oh.
They have to inhibit microbial growth.
Oh.
And they put nitrites in wine. And some people are allergic to them. But they're preservatives.
Oh, okay, thanks.
Now, what we've just seen here, reducing nitrate to nitrite to some type of gas, that is one metabolic pathway for recycling nitrogen. There are actually 4 major pathways, and they're used by different microorganisms. We have nitrogen fixation, ammonification, nitrification, and denitrification. but it's the microorganisms that do all the nitrogen recycling on the Earth. And so where do we find the majority of nitrogen on the Earth?
Is it in the soil?
Nope.
In the air?
Yep, in the atmosphere. Remember, the atmosphere is mostly nitrogen gas. And nitrogen is one of those essential elements, right? We need nitrogen to make amino acids, to make proteins. We need nitrogen to make nucleic acids, right? Those nucleotides. So we need nitrogen. It's essential. So where do we get it? Do we simply inhale deeply and get all the nitrogen we need there to make our proteins and nucleic acids? Where do we get our nitrogen?
Oh, I think I got confused. You get it from the soil, right? From the plant?
Hmm.
And from the
Well,
animals?
but humans, where do we get our nitrogen?
From eating?
Yeah, from eating, right? We eat. We eat animals, we eat plants, that's how we get our nitrogen. So remember, plants are the primary producers, the base of the food chain. So how do plants get their nitrogen? Now, Rachel.
From the soil?
Right from the soil. So, but how does nitrogen get into the soil? Well, it needs the help of microorganisms, right? So when plants and animals die, microorganisms break down those organisms, right, to put nutrients back into the environment where they can be reassimilated. But what if you have a plant and it's growing in a very poor soil? So there's very little nitrogen in the soil. Well, we have certain plants, remember I talked about these, that have a bacterium that infects their roots. And that bacterium was rhizobium. And rhizobium can perform nitrogen fixation, where the rhizobium bacteria absorb nitrogen from the air and convert it or fix it into compounds like nitrates. So the bacteria make the fertilizer for the plants. So it's the bacteria and other microorganisms on the earth that recycle all the nitrogen, so that it is available for plants and animals. So we are entirely dependent on the microorganisms, but they're not dependent on us. We need them, but they don't need us. But without microorganisms, there is no nitrogen recycling. And this also occurs in water, in the ocean, lakes, streams, ponds, because there are plants and animals that die and decompose, but also the fish and other aquatic life, they produce a lot of ammonia in their waste. So if you've ever had an aquarium where you kept fish, you know about this. For those of you who have never had a fish aquarium before, let's say you want to get one and you're all excited. So you go to the fish store, you buy your aquarium, you buy your rocks and gravel, and you buy some fish, not goldfish, but other kinds of fish. And so you fill the aquarium with water, you add your dechlorinator, and you put all your fish in, and the next morning they're all dead. And they're all dead because they poison themselves with ammonia from their waste. Because what you have to do when you're setting up a fish tank, whether it's freshwater or saltwater, you have to put things in and then maybe put in like a goldfish, which can live in crap, so it doesn't care. But you have to give the aquarium time to acquire a population of bacteria that will break down the ammonia. And in that pathway, it goes from ammonia to nitrite to nitrate, which is the least toxic form. And so if you don't give your aquarium time to build up that population of bacteria, then all the waste from your fish will kill them, will poison them. So you usually start out with maybe one fish, like a goldfish, which, as I said, it can tolerate almost anything because it breathes air. But with other fish where the water has to go across the gills, if there's a lot of ammonia there, it's going to kill them. And then over time, you can start adding more and more fish, right? Because the aquarium now has a good population of bacteria to break down the toxic ammonia into ultimately nitrates. And then you do periodic water changes, right, to remove the nitrates and keep everything nice and happy and healthy. And they do sell test kits where you can test for nitrate and nitrite and ammonia and stuff. And the test is actually based on what we're doing here. They use sulfonylic acid, alpha-naphthylamine, and zinc. And you get a color change, and they usually give you a little card with different shades of that color so you can compare to see what your nitrate level is and things like that. So this is a common test. It's also commercially used, you know, for people who like to keep fish in aquariums and things like that. But all bodies of water have these microorganisms to break down all that toxic ammonia and everything. So, you know, bodies of water are dynamic ecosystems. There's a lot of chemistry going on along with all the life and everything. So it's something to think about, that it's all the microorganisms on the earth that recycle all the nitrogen, that break down things, make the nitrogen available for everything else to use. And microorganisms, they can create nitrogen gas, right? They can break down the nitrate and convert it into nitrogen gas. Or they can do the opposite. They can take nitrogen gas and convert it back into nitrate. So all the nitrogen recycling on the earth happens because of microorganisms, and we are dependent on them, right? Because of that, because we cannot inhale our nitrogen and use it. We have to get it from the foods we eat. And remember, the food chains start with plants. So plants have to be able to absorb nitrogen from the soil or from bacteria living in their roots. And then animals eat the plants, other animals eat the animals that eat the plants, and so on and so forth. So, any questions on nitrogen recycling? So one of the gases produces nitrous oxide. Are you familiar with nitrous oxide? Nobody's gotten high from nitrous oxide? You can go to the dentist and be given nitrous oxide, right? Makes you, yeah, laughing gas, right? Makes you kind of loopy and kind of euphoric. It is safe, at least under those conditions. Actually, nitrous oxide has an interesting history. When it was first developed, it was actually used by, you know, rich people, the aristocracy, to have nitrous parties where they would all go get high, but only rich people could afford it. And then later, it was actually used as an anesthetic, like at the dentist, you know, to help relax you. It's used in your whipped cream that's in a spray can. Have you ever bought the whipped cream in a spray can? They actually put nitrous oxide in the can to help preserve the whipped cream and to help it spray out. So that's nitrous oxide. Unfortunately, nitrous oxide, which is produced by soil and water bacteria, so there's a lot of natural nitrous oxide produced, but not in high enough concentration for you to get high by taking a walk or a swim or anything. But nitrous oxide is actually a very powerful greenhouse gas, much more powerful than carbon dioxide is. So there is a downside to nitrous oxide. It is a greenhouse gas, but it's naturally produced. And the Earth could deal with that if not for humans burning fossil fuels and putting a lot of CO2 in the atmosphere. But nitrous has an interesting history. So any questions or comments?
I don't think the laughing gas works so well. I've had it before.
Well, it's not really that good for pain reduction. It's more just to make you calm and relaxed and not that concerned about what's going on in your mouth. But no, for pain, you really need an injection of anesthetic, right? So you need that in order to eliminate pain, you know, the Novocaine. But laughing gas, I've only ever had it once, a long, long, long time ago. I don't really remember that much about it, but it didn't smell very good to me. So That was probably the rubber mask that, that smelled funny. But yeah, it's mostly just to relax people, who might be nervous. But you can buy nitrous. It's legal. At least for right now it is. So any other questions or comments? So what are we doing in lab next week? What's happening in lab next week?
Did you say you were doing the unknown, the second one?
Well, that's the week after next.
Oh.
So next week is all biochemical tests. So we're going to be learning a lot, and yeah, probably street plate in there too. But a lot of biochemical tests because we need to learn all those because that's what you're going to use on your second unknown. So we're going to be doing a lot of biochemical testing. So there's a lot of different tests. You need to understand how the tests work, what we're testing for, the kinds of organisms, that give a positive or negative test result. So it is a lot of stuff, but you guys don't actually have to perform any tests. We're just going to talk about them and look at their results. And then for your unknown, you just look at pictures of the results and interpret them rather than actually inoculating and incubating and doing the test and getting the result and then putting it all together to identify your unknown. So the remote unknown is actually simpler, right, than what you would do in lab. So anything else? All right, that'll do it. It's supposed to be a beautiful Halloween, just a little cool, so you can enjoy your trick-or-treating.